thank you for having this lecture today. The microphones are yours.
Thank you. Thank you, Omar, for this very nice introduction. So, what I wanted to do with you today is to share some of my fascination for the little organ called the pancreatic islet, or the islet of Langerhans. And I want to go through and discuss with you, and of course, you can interact, ask questions, whatever you want at the end of the day. Where you're getting. But I will first give you some sort of a motivation to why we are interested in this. So you know, and this is a very, very difficult situation also in Mexico, where you have a huge population of obese type 2 diabetic people. And diabetes is mainly due to the fact that this little organ, the islet of Langerhans, is not working properly. So if you look at human physiology, these islets are presented within the overall exocrine pancreas, constituting about 1 million in the human pancreas. And the total volume of all of these islets is just 1 to 2%. So there is a huge portion of this organ that has nothing to do with the pancreatic islet. If this is not working, if insulin release is not working, you get all of the complications that I probably do not need to talk to you about in the vascular tract, in the nervous system, leading to severe complications like blindness, like kidney failure, and if really severe, to amputations of legs, etc. Within this little islet, there are a number of different cells that are working in concert to regulate blood glucose homeostasis. And the cell that we have been focusing on a lot is the insulin-secreting pancreatic beta cell. So this is a schematic picture of the pancreatic islet, where you have insulin-secreting cells, you have other hormone-secreting cells. What you also can see that there is a rich capillary network within these islets, and they are also exposed to various types of innervation. So this is where this little microorgan is getting complex, because you have autocrine feedback loops, which means that things secreted from the pancreatic beta cells, for instance, are regulating in an autocrine fashion signaling in the pancreatic. But there is also paracrine signaling from the other endocrine cells. And there is endocrine signaling, which means that factors are coming through the bloodstream to the pancreatic islets. And all of this signaling needs to be integrated at this little organ level in order for it to work and function in an appropriate way. So I will not go through in detail the pancreatic beta cell here, but this is what we have been working on for a number of years, trying to understand when blood glucose concentration is changing, what happens with glucose uptake to the cell, metabolism of glucose, generation of different types of signals, closure of different types of ion channels, opening of other ion channels, that all are then aimed for regulating release, secretion of the insulin containing obesity. And you can also see that in addition to channels and transporters, there are a number of receptors in the pancreatic beta cell plasma membrane. And again, at the cell level, the cell is integrating now signals coming from all of these places that I told you in the previous slide. And this is the fundamentals, the basics for functioning of this little organ. So here you see another picture of the islets, actually a reconstitution of micrographs, microelectronic graphs. And you can see here the whole organ with the blood capillaries, with the white nerves, and with the different cell types, and how this little beautiful organ is looking like. And I just want to illustrate once more that this is a complete organ. This is a very well-defined microorgan, and of course, it can be used for a lot of different types of studies, not only diabetes. So if you have an interest in cell signaling and want to understand how cell signaling is regulated at the cell and at the organ level, you have the possibility to look at an organ like this. What is also important to illustrate, and you know it very well, that for a lot of our studies in the laboratory, we use experimental animals. We use, for instance, mice or rats, etc. But you can also see here that the architecture of these islets between mouse and human are very different, whereas human and monkey are very similar. And if you look here at the different cell types, so in this case, the red cells are the pancreatic beta cells, the insulin secreting cells. You can see that they are localized within this islet in a much more mixed pattern than in the mouse islet. And it's not only that this reflects an architectural difference, that there is also a functional difference. If you look here at the mouse islet, you find parasympathetic fibers, and these fibers are important because they release acetylcholine that is enhancing the sensitivity of the pancreatic beta cell to glucose. In the human islet, you do not see these type of parasympathetic fibers, but still acetylcholine has a very important role in sensitizing the insulin secreting cells to the prevailing glucose concentration. But in this context, the alpha cell, the glucagon secreting cell, is also secreting acetylcholine that then acts on the beta cell in a paracrine action. If you then look at sympathetic innervation, you can see in a mouse pancreatic islet, the sympathetic fibers reaches the endocrine cells, whereas in the human islet, the sympathetic fibers reach the smooth muscle cells of the capillary and can then regulate blood flow within this islet. So if you now take a bigger mission and you want to design drugs to treat diabetes, you need to be aware of that the mouse models are not always telling you the truth for the human situation. So when you are looking for pharmacological treatment strategies, you need to validate that what you see in the mouse model also holds through in the human being. And one step is in between is the non-human primate, which is looking very much like the human. You saw it in the human islet comparison. So, back in 2006 and 2007 or so, we got irritated on ourselves because we said, that we are taking out this little microorgan, we are dissociating into cells, and then we look at signal transduction events in detail, trying to understand how this complex machinery is working. But this is in vitro, this is outside the body. And of course, that is a very, very oversimplification how things look like under real conditions. It's like taking out a part of the car engine and then try to understand how the whole car is working. So we understood that we could not go on doing these type of things. So then we decided that we should transplant pancreatic islets into the anterior chamber of the eye. And the reason for this was that we could then use the cornea as a natural body window for imaging. And now you can use whatever type of microscopy that you are interested in, whatever type of sensors, fluorescence-enabled sensors that you are interested in. You can image non-invasively, longitudinally at single cell resolution for very long time. And here you can see the individual islets in the mouse eye. You can see the capillaries. You can also see that some of these islets are clumped together, and you can see the pupil of the mouse eye. What is interesting is that these islets, they are living in the eye for very, very long time, and they are not changing their architecture, so they are there. So that means that you can transplant a young mouse, and then you can follow signaling over many many months. So basically for the entire lifespan of this animal that you have put here. What is interesting to note is that these islets are getting both vascularized, the black circles here, and innervated, the red and the blue circles. And you can see that the nerve fibers are finding their ways into the islet alongside the capillaries. And what is important to note is that this takes about 30 days to have full vascularization and full innervation. The vessels that are formed, so this is the pancreas, the normal place where you find the pancreatic islets. And there you can find in the vessel wall, the capillary wall, you have very nice small openings, fenestra, that are allowing a very efficient exchange between the blood and in this case the pancreatic beta cell. You see the insulin stored in small vesicles. If you now transplant your pancreatic islets to the anterior chamber of the eye, you can see that you have the same type of openings, enabling the same type of very efficient exchange between the blood and the cell. However, we were somewhat surprised because there is a trick. You can make a mouse very diabetic with injection of a chemical called streptozotocin. And if you do that, you can see that the blood glucose concentration goes up tremendously. Now, if you transplant pancreatic islets into the anterior chamber of the eye, you can see that you get normal glycemia. And that normal glycemia lasts until, in this case, we take out the islets from the eye after about 250 days in this context. Then hyperglycemia comes back. And if we do a glucose tolerance test, the same thing as we do in patients, you inject, so this is the diabetic patient or mouse, you inject glucose, and then glucose goes up, and then you measure the time it takes for the glucose concentration to go down to normal values. And you see, in this diabetic mouse, nothing happens. This is a normal mouse, the black circles. You inject glucose, glucose comes up, and then it comes down over time. If you now take this heavily diabetic mouse and you do transplantation to the anterior chamber of the eye, you can see that we have virtually a completely normal glucose homeostasis. So you can treat diabetes by this islet transplantation to the anterior chamber of the eye. So this is what we call a metabolic transplantation. We replace the pancreas with pancreatic islets in the eye. But we can also use a reporter concept, which means that we put just a few pancreatic islets into the anterior chamber of the eye. And then, of course, we can image these islets over time using the cornea as natural body window, as I said. And then these islets will report on what is going on in the pancreas. And of course, here we cannot image anything. Here we can image with cell resolution. And whatever changes we see here are reflecting changes that are going on in the pancreas. So you can also use this for autologous transplantation. So in this case, we were interested in looking at certain aspects of signaling in monkey pancreatic islets. So what we did, that we took away about 20% of this pancreas of this monkey. We isolated pancreatic islets in small microorganisms. We transplanted them back in an autologous fashion to the anterior chamber of the same monkey. And then we were able to image whatever we were interested in imaging. Importantly, thus, this type of pancreatectomy does not affect body weight, does not affect blood glucose, and does not affect fasting glucose. And what you can see here is a few islets, higher magnification here. So this is now a reporter transplantation where we wanted to read changes in the pancreatic islets in this particular monkey. And in this case, we were interested in blood flow regulation. We can also use this approach for human pancreatic islets. So we can transplant human pancreatic islets into the anterior chamber. And here we now use immunocompromised mice, which are not reacting to this transplantation. And we make these immunocompromised mice diabetic by streptozotocin. which now means that the only islets that can handle glucose concentration in these mice are now the human islets placed in the anterior chamber of the eye. And what you can see here is that the blood vessels, the capillaries that are formed within these islets from the human that are red, staying here, they are reaching full vascularization after about 30 days, exactly the same as I showed you for mouse models. So now vessels from the mouse are supporting the human pancreatic islets with blood flow. And this way, the blood going through these capillaries are very effective in giving the nutrition, oxygen, etc., to the human islet. What is then interesting to note is that now you have human islets in a mouse. And if you look in a human, you can see that the non-fasting glycemia is less than that of the mouse. And that is true also for the monkey. So in this experiment, what we did was that we took out the endogenous beta cell in this mouse with streptozotocin. Then, of course, we got hypoglycemia. Then we transplanted to the anterior chamber of the eye these islets from mouse, from human, and from monkey. And what is quite remarkable is that if you look at the human islets, so the human islet is restoring the glucose concentration in the mouse to the same level as was in the human. So this means that these islets, they are serving as glycostats. They are determining what type of glucose concentration does the human islet want in order to function properly. Of course, you can also use this for other approaches. You can use it for drug screening. So if you put human islets into immunocompromised mice, as we call it, then a humanized mouse model, and then look at the human islets in the eye, you can test different types of drugs, and you can identify novel druggable targets and test drugs for these targets. And I just wanted to emphasize that it's not only human islets that you can put in here. You can put in anything, spheroids or any organ that you're interested in. You can put in organoids from different stem cell derived lineages that you're interested in. And the beauty is that you have now a body window where you can follow these organoids, spheroids, islets for a long time at the single cell resolution. So this worked so beautifully. So we also actually got the permission both from the FDA in the US and the regulatory authorities in Europe to do transplantations to patients. And this is actually something that we are now going to set up here at Tec de Monterrey at the Zambrano Hospital. And we are very excited to do this. And in this context, we will, of course, go for the very large type 2 diabetic populations. And we can go into that a little bit more if you're interested. But we identified these patients and we transplanted this patient exactly the same way as we did in mice and in monkeys with pancreatic islets to the anterior chamber. So if you are now interested in looking at different types of signaling, I will give you a few examples. So one important signal for pancreatic beta cell function and survival, or for cell function in general, is cytoplasmic free calcium concentration. And you can actually transplant these islets into the anterior chamber of the eye and then use a sensor for changes in cytoplasmic calcium. What is important to realize here, and there has been a lot of studies done in vitro, but really very few studies in vivo. And we started to do these in vivo studies because, again, we wanted to figure out how it looked in real life. And what you can see is that it's not a homogeneous number of cells within this pancreatic islet. They are responding differently to glucose, and they're also showing a difference in basal activity. And what is important is to understand then in this context the interplay between different types of calcium channels in the plasma membrane, and calcium mobilization from intracellular stores like the endoplasmic reticulum. Because these two pathways are leading to the complex, intricate, and important dynamics of calcium patterns within these cells. And these dynamics of calcium patterns are important for exocytosis, for secretion, but they are also a very good reporter for cell function. And if you record these type of patterns, you will see that they are showing oscillations in cytoplasmic free calcium concentration. And you can see changes in these patterns in different cells, but moreover, you can also see changes in this pattern if the cell is not happy, if the cell is stressed, if the cell is old, and if the cell is diabetic. So this is an important tool that we have used and are continuing to use in understanding how exactly these patterns can tell us about cell function and survival. During these type of studies, and I said that the anterior chamber of the eye is a beautiful in vivo, giving us a beautiful in vivo imaging. But what one needs to keep in mind is that when we're doing these recordings, these are then pancreatic islets put into a mouse that is under anesthesia. And you can see here that an example of two types of anesthesia, isoflurane and hypnon. And if you take isoflurane, for instance, as an anesthetic, you can see that we have very large calcium oscillations in the cell. However, they are completely disconnected with the insulin secretory apparatus. So these calcium changes cannot be converted into signals that will lead to insulin release. Whereas if you use another type of anesthetics, you can see that you are still maintaining calcium oscillations, and in this case, they can be directly linked to activation of the insulin secretory process. So these are things that are important to keep in mind when this type of experiments are done. So for that purpose, we wanted to have the ability to do recordings with high stability, with high resolution over time in awake animals. So what we then did was that we were transplanting pancreatic islets onto the dura mater of the brain, where they get very nicely, again, vascularized and innervated. And what you see here is a cranial window with the islets on the dura mater. And in this case, we are not putting these mice under anesthesia, but we are actually allowing the mouse to move freely. And it's called metabolic or mobile home cage for these mice. And in these ways, we have now an opportunity to do certain aspects of signaling under completely normal conditions. And that, we believe, is a very, very big and important step forward. And here you can, for instance, see recordings of calcium changes and recordings of insulin secretory cells. So this is where we are now going to combine these two in vivo approaches, trying to get more of an authentic picture of how signaling is regulated within the islet and the islet cells. I told you that you could put islets into the eye and they will be there until we take them out. That is not really true. That is dependent on the syngeneic transplantation, the same type of mice. However, if you do an allogenic transplantation, different strains of mice, then you have a reaction. And the reaction takes place after about 30 days. You remember that 30 days was the time it took for full vascularization to take place. So although the eye is immunoprivileged, as soon as the capillaries grow in to the islet, you break that privilege. And then you get a full-blown immune reaction and the islets disappear. That you can, of course, use to your advantage. So again, here is now an islet in transplanted into the eye of a mouse, and we have labeled, in this case, the T cells, the immune cells, with GFP. And you can see here how these T cells are now attacking in real time the pancreatic islets in the anterior chamber. And you can also see that these T cells represent different phenotypes. This looks different from this one, and this looks different from the other two. And if you look more in close contact, you can see that our round T cells, elongated one, and roughened ones. And with the resolution we have in the eye, we can actually look at the movement patterns. So the round ones are not really moving around. The elongated ones are covering large trajectories, whereas the roughened ones, and the reason for calling them roughened is that they look roughened, they are moving, but they're not covering the same ground as the elongated. What is interesting is at the peak of reaction, it's actually the roughened cells that are the most important. And what happens when these roughened ones are attacking the pancreatic beta cells? You can see a few of them in action here, again in the eye. And what happens is that these roughened ones, they release enzymes like perforin and granzymes that are poking home on the, in this case, grayish, blackish pancreatic beta cell. So one of these roughened ones can kill about 70 beta cells, insulin secreting cells, in 48 hours. So it's a very efficient. And of course, now you have the possibility to intervene either systemically with different types of immune suppressive drugs, or locally, directly with eye drops or eye injection, and then start to understand what type of reactions that are going on at the molecular level, and then see how you can intervene, which is clinically important. What you also can do with this little microorgan, you can dissociate it into the single cells, and then you can transfect this with different types of constructs that you're interested in getting into your pancreatic beta cells. And then you can reassemble these cells into pseudo-islets. And these pseudo-islets, you can actually determine pretty much how big they are by using different numbers of cells. So you can produce bigger ones or smaller ones. And these pseudo-islets that you are now creating are behaving completely normally in terms of glucose stimulation. So here you have a pseudo-islet that you stimulate glucose. In this case, you measure calcium. Calcium goes up, shows an oscillatory pattern. So we were very happy to see that we could now transfect all islet cells and then create these pseudo-islets for further experimentation. And what you can see here is that when we place these pseudo-islets on the anterior, in the anterior chamber of the eye, they are looking very nicely. They are engrafting onto the iris, and they are vascularized again with exactly the same type of vessels that are occurring or are existing in the pancreas, with small openings enabling this very important exchange. And if you now transplant these pseudo-islets into a diabetic mice, you can see that they are regulating glucose concentration. So now we have created a situation where we can express different types of proteins within the islet and actually understand in detail what is going on. I mentioned to you that you also could do organoids from different types of stem cells. So we have been interested in creating our own differentiation protocol to get to insulin secreting stem cells. And obviously, from a research point of view, from an experimental point of view, this is very interesting. But it's even more interesting when you start to talk about clinical islet transplantation, because we will never ever have enough islets coming from cadaveric pancreases, and cadaveric pancreases are donated to us by traffic accidents, other type of accidents. But if we are looking now at the number of people having diabetes, so 2030, we predict that 600 million people have 600 million. Simultaneously, there will be around 300 million that not yet know that they will get diabetes. So this is a catastrophe. So something needs to be done. And I'm personally a big believer in regenerative medicine. In other words, that you replace organs that are not functioning. So in order for us to get a therapy, a valuable therapy going for patients with diabetes, both type 1 and type 2, we need an amount of islets that we can transplant. So we have high hopes for stem cell-derived islets. And there are a lot of protocols out there. There are a lot of people that have looked at this, but they are not very convinced and not very happy with what we have seen. So we did this protocol ourselves. And obviously, these islets need to produce insulin, but that is not so difficult. But they need to produce the whole sensitivity apparatus for glucose. So they need to be sensitive to the small changes in glucose concentration in the blood, and always being able to adjust their secretory capacity to that change in glucose concentration. And that sensor mechanism is not that easy to get. So that is one problem. The other problem that is very important is, of course, that if you do these stem cell-derived islets, they cannot contain undifferentiated cells. They cannot contain non-wanted cells, and they need to contain only mature pancreatic beta cells. If not, it will essentially be that you are transplanting a tumor into the patient. So when we were doing our differentiation protocols, we transplanted these pancreatic islets, stem cell-derived islets, into mice that were diabetic, and you could see that they were functional. We could adjust the blood glucose concentration by these islets in the same diabetic models that we described before. And of course, these are human material. They are human cell lines that we created these stem cell-derived islets from. So in order for us to be able then to measure insulin release, we measure a proxy for insulin release, C-peptide. And as you can see that in the same time interval that reduced the glucose concentration in the blood, we have a production of human C-peptides in the blood of these mice. If we did a glucose tolerance test, the same as I described to you before, you can see that there is an in vivo maturation after transplantation. So after 6 months, you have very nice and effective glucose homeostasis. And again, after 6 months, you have a very nice production of human C-peptide. And importantly, if you now look here at transplantation, you have the insulin, you have glucagon, and you have another hormone that I didn't mention, somatostatin. But these are key hormones, produced by key cells in the pancreatic islets, and they are all there. And these stem cell-derived islets are very nicely vascularized. And if you look close on this immunohistochemistry, you will not see any exocrine cells, you will not see any unwanted cells, and you will not see any proliferative cells. So this is a very good sign of that we may have here something that could be developed further for clinical transplantations. What you also can do, of course, is that you can use pharmacological intervention. And we talked about the immune reactions, and I said that they are tough, and you may want to test different ways how to intervene in an immune reaction. So in this case, we did transplantations of small, chemically defined particles that were not loaded or loaded with an immunosuppressive drug. In this case, we used rapamycin. Then we transplanted in these particles together with the islets to the anterior chamber of the eye. And what we could see, and in this case, we decided to have these particles slowly degrading over 30 days. And in this case, we could see that if you had nothing, no immunosuppressive drugs in the particles, all of the islets were gone after, in this case, 30 days. Whereas all of the islets remained after 30 days if these particles, while degrading, were releasing this immunosuppressive drugs. You can actually see here in a scheme that if you have rapamycin present, you delay the destruction of the pancreatic islets. And of course, here you can now design particles that are released even slower. And of course, the hope we have is to be able to tell these particles when they should release. And you can think about radio waves, you can think about magnetic fields, etc. But here you have a direct possibility to use pharmacological intervention in the anterior chamber of the eye to be together with what we have transplanted. So another aspect that we are very excited about, that is actually to be able to measure things in the eye. And I gave you the example where we developed the mobile home cage, not having to use anesthesia. But of course, the dream would also be to create something that can be used for measuring what's going on with the islets in the human eye. Because, again, having 600 million plus 300 million with diabetes, there will not be enough doctors or nurses to take care of all these diabetic patients. So there is a wish, of course, that each patient could be their own doctor or their own nurse. And in that context, being able to judge what is happening with the transplant. So in a very nice collaboration with the Royal Technical School in Stockholm in Sweden and another technical school in another university in Sweden, we were developing a small unit whereby we could put the islet inside. And this unit, of course, have a lot of different abilities. So it was equipped with an LED light source. It was equipped with a photodiode that could sense light that were sent out. It was a little small computer that could store signaling. And it also was a device that allowed sending signals, light signals, from the emitted light by Bluetooth to amplifiers outside the eye. And this is something that we haven't gotten to the extent that is done yet, but we have really created this small device very successfully. We use a lot of microelectronics that is needed for a lot of different purposes. And of course, a lot of these things we needed to develop ourselves. But this is where we hope to be able to go in a not too distant future. And obviously, we are developing this small sensing device for the eye. But if successful, needless to say, this small sensing device could be put anywhere in the body to perform these types of things. So our directions here for the future is both in Sweden and elsewhere, but also here at Tech de Monterrey, is to move forward with this project and actually doing it with our two arms. Basic science, which means looking at signal transduction of these islets, the different islet cells, and the different subcellular compartments. And of course, translate these findings that we have into real life in a translational research project. Learning more how the beta cells are working and why they are not working in diabetes. And also using this modeling system for target identification and testing of drugs for these targets. So this will basically be done in mice and humanized mice. And obviously, what we want to do is then to take islets that we genetically manipulate prior to transplantation. Because you can do all sorts of genetic manipulation there. You are doing it outside the body, and then you transplant that genetically changed material into this mouse. But we, of course, also want to move with the human islet transplantation. And here, of course, is a lot of stem cell-derived islet characterization needed. So what we want to do is to take these islets or stem cell-derived islets, transplant them into the anterior chamber of the eye of both type 1 diabetics, which we do in the US and which we primarily do in Sweden and in Europe, and type 2 diabetic patients, which we will be doing here primarily in Monterey. And then, of course, look at the outcome in this type of clinical studies. And in both of these arms, we are, of course, heavily dependent on imaging system, but also pharmacological treatment strategies that we deliver into this anterior chamber of the eye. So this is how we see future research going on in diabetes that we believe is very important. And I think that it's absolutely necessary to move from the in vitro stage into the in vivo stage, because again, the complexity that you have in the body, whether it's a mouse or a human, is enormous. And in order to identify these small changes, the signaling things that are happening, is need to be in the vivo situation. And there are lots of other things that we want to move. We want to move with higher resolution microscopy. We want to move with the, and in this case, with quantum sensors, higher resolution sensors, better stability, maybe fluorescent probes in the near infrared range, not distracting, not disturbing the tissue, which we may then be able to take into the human situation when time comes. Okay, so this is just the lab we have now in Stockholm. And we have a very exciting, great possibility for collaborations here in Monterey, and I'm very happy to be here. I like Mexico a lot. I think it's a fantastic country to be. I see a lot of possibilities for the future here with the research we're doing. A lot of very talented students, postdocs, etc. And tech is a fantastic word with a lot of giving us a lot of possibilities. Now I have talked enough, so please, questions.
You for this wonderful talk. So now I'm open for the audience to ask questions